Okay, so this session is called Making Measurement Meaningful. And that's not to say that the last few sessions have been about meaningless measurement. In fact, it has been genuinely really heartening to see how much focus there's been on making sure that any monitoring that we're talking about or biodiversity measurement is useful and crucially can be used. But as a number of speakers have pointed out, we're in a situation at the moment where we've got more biodiversity data than we've ever had before and way more sophisticated tools than we've had before for measuring and monitoring biodiversity. But I think everyone in this room would agree that that richness is not doing as much as it could for tackling the biodiversity crisis. And that's the purpose of this session. And we've got the most fantastic range of speakers with a really diverse skill set. So I'm super excited about the talks. First up is Nick Slavsky. He is Executive Director of Foundations of Success. For the last 25 years, they've been working on trying to make conservation practice more effective. So, Nick, over to you.
Do we have the clicker? Clicker?
It should be there. It'll walk away. Yeah, that's it.
Oh, that's it. Okay.
That's it. Yeah.
Okay.
The green.
Okay. Thank you. All right. And if I could get the slides. So thank you very much. This is a practical approach to measuring biodiversity, transforming monitoring data into management information. A little bit of, so I represent a number of different groups, the Conservation Measures Partnership, which is a community of practice of all the organizations that you see here. We create the open standards of the practice of conservation, which have gone through now 5 different iterations over the last 25 years as a set of best practices for doing conservation work. I work on Marathi software, which helps people implement those standards. And I'm part of the IUCN World Commission on Protected Areas, which is helping measure 30 by 30. So a lot of this comes from that experience of working with practitioners to do better work. I want to start with an analogy. We talk a lot about gauges, and I want to talk about as an example of gauge you're familiar with, which is the fuel gauge in your car. What makes that fuel gauge useful? I would submit it's 3 things. That there's an information need. You're making a management decision. When do I pull over and get more fuel? There's an actual measurement. So that needle measures the amount of liquid in your tank. Everyone fixates on the indicator, but that indicator doesn't mean anything absent the markings that say full, half, and empty that help you interpret what that indicator actually means. It also, if you get smarter, you can actually get to the information you actually need, which is that. And one of the things here is it doesn't really matter if that's super precise. It just needs to be accurate. We need to know that we have about a gallon of gas or about 4 liters of gas left. If you look at cars, we also build gauges like this, a tachometer, which supposedly has an information need. But if you drive an automatic, or even if you drive a manual and you do it by ear, you never use this thing. So it's a waste of cognitive space and money to create a gauge we're never going to use. So we don't want those. All right. So what I want to talk about is focal conservation values and viability analysis, proven tools to construct useful biodiversity gauges. And this is based on academic literature and a lot of practice going back 25 years and more. So our problem is we're faced with that. We have this system out there and we need to make sense of it. How do we get our heads around the thousands of species, the dozens of ecological processes going on in that space? What we've learned how to do is to select what we call values, which can be ecosystems or they can be species. So you start with the ecosystems that you care about, and then you think about, are there any species that would fall through the cracks? And we're trying to pick a handful of nouns, of things that for us represent that overall system. So in case you can see what we've picked in this case. Once we have that handful of nouns that we've picked, then I'm going to give you an example using wild dogs in Africa. We would pick as our focal values a painted dog population, the prey population, and maybe the savanna ecosystem where they're working. What we're then going to do is we're going to think about key attributes of each of those values. So maybe successful breeding units is something that we want to care about for the painted dog. And for that, we are going to come up with an indicator, the number of packs that raise pups to adulthood, And for that indicator, we are going to create the markings of poor, fair, good, and very good. Where do those markings come from? Let's say we want to have fair is 5 to 15 packs. Greater than that is 15 to 20 is good. So we're kind of looking at those markings that we have there. Any natural indicator is going to go through variation over time. We already saw that in this morning. And the key here is to define what is that natural range of variation. So when are we within that natural range of variation or acceptable range, and when are we outside of it? And then that key threshold becomes that marker between fair and good. I notice my lesson sign is reversed here, but anyway, you have that key threshold between fair and good. So that good is within the acceptable range of variation, but you might need to do something to keep it there. Fair is when you're outside that range of variation and you require intervention to get back to good. Poor is you're likely to go extinct, and very good is you're in an ecologically stable state. So we're defining what those markings are vis-a-vis that indicator for the thing that we care about. And then we can set where we are today, and we can also set a goal for the future. This can be done quantitatively. So here's another example with toads that maybe have in ponds, and they have to hop from pond to pond. So we might quantitatively assess the number of ponds with egg masses, but we can also do these assessments qualitatively. We don't always have to be super precise, but as long as we're conceptually accurate, well, how many ponds have a fungus present? Many, few, or none. Or how easy is it for the toads to hop from one pond to another? It sounds silly to construct a hoppability index, but that's what we actually need to know to manage how these toads are doing. So what I've showed you is how you might think about that at a simple scale of a small project or site, but we've been doing work with the Jane Goodall Institute with chimpanzees in Tanzania, and we can actually start to apply this to all the other data that's out there. So as you know, there's a torrent of data out there, but not a lot of information. So we have things like satellite measurements of pixels of trees. We have drones surveying chimpanzees' nests. We have community surveys or smart things tracking snares in the forest. So we have all this data that's out there. How do we make sense of it and give it to decision makers? We need to take those pixels of data and convert them into meaningful management information. So the traditional way of doing that would be to say, well, here's some areas where the chimps are. How's their habitat quality doing? And we'll give it a kind of a rough guess of the quality in terms of those 4 colors. Goldilocks would say, yeah, that's far too imprecise. That's not very helpful. But if I just give you all the pixels of the trees that are out there, Goldilocks would say, it's too detailed. As a decision maker, I can't make sense of all those pixels. So what you can do is you can construct one of these viability tables around those remotely sensed data or the different sources of data that you have. You can create those buckets of poor, fair, good, and very good. And based on those buckets, you can then assign management polygons the status of how they're doing over time. And that's something that decision makers who are managing those polygons can actually use. And Goldilocks would say, that's just right. And we could even then extend that, if you want, to all of Africa. So we can extend that same kind of logic and thinking to making sense of all that data that we have coming across Africa to say, how are chimp habitats doing overall? So the key words for this viability assessment that I want to leave you with is that it's representative and encompass. That we're picking a handful of indicators. If they're okay, our attributes are okay. If they're okay, our conservation values are okay. If they're okay, biodiversity as a whole is doing okay. We are constructing a model. And as we all know, all models are wrong, but some are useful. And I think that this is a really useful model for decision makers. If you go back to the criteria for a good gauge, it has an information need. So let's go back to that toad example, the status of the toad species. We need to know that for whatever reasons. What's the actual measurement, the hoppability index? And how do we interpret that? If it's hoppable, then we're fine. If it's not hoppable, we have to take management action to change that world. So again, we're trying to construct a gauge that is useful for decision makers. The last thing I want to leave you with is, I know this is about biodiversity, but the conservationist in me has to say, we also have to think about other indicators of success as well. And I'm going to give you one more example here. Let's pick 2 identical pieces of forest out there in the land, Site A and Site B. Site A has humans in it and they hunt throughout it. Site B has never been touched by a human being. No human being has ever set foot in that. I'm going to ask you, which of those 2 sites is better conserved? And if we go on purely biological data, we would have to say Site B. If we're going to really say that all the species, all the ecosystem functioning is totally intact in that system. but you know it's a trick question. And if I give you one more piece of information, which is 6 months from now, a large logging company is slated to come and clear cut that piece of forest. But remember, it's 6 months before, and I ask you which is better conserved. I think we would all say site A is actually better conserved. And what that teaches us is if we really care about biodiversity, yes, we do have to measure the ultimate outcome. Yes, we do have to measure biodiversity. But we also have to track threats and how threats are doing. They're a really important leading indicator rather than a lagging indicator of how things are going. And we have to track the capacity of humans and people to respond. And if we really want to talk about success in measurement, we're going to have to create the system to look at all of those different things. So thank you very much. These are some of the things and resources you could get to learn more about this.
Fantastic. Thank you so much. Questions for Nick?
Thanks. That was a really cool talk, and I'm super excited to hear all this discussion around actionable data and decision appropriate data. And my question is really to the kind of end interpretation of those ratings you talked about. I think in the examples you gave us around the chimp habitat, it was clear kind of you went from the low, medium, high, or whatever, but in a real decision context, the differences between those categories often have real world consequences for like go, no go in terms of investment or other things like that. So how do you deal with just both the challenges of that interpretation and then finding kind of meaningful thresholds without kind of getting lost in that kind of deep need for more and more data analyses? Yeah.
So that's a really great question. And it comes to this point that we discussed, some of us this morning, about the role of science and scientists in these processes. Scientists, I don't think, can make the decisions, and I think our credibility depends on us being impartial. But there is value even in choosing what threshold to do and how to assign those ratings. And I think we cannot shy away from that. That our role is to use our ecological knowledge to help define those polygons and those colors for those decision makers so they can make those decisions. And yes, there is bias, and yes, there is human context in the way those are set. But that's the way the world works. And we have to convert those pixels into polygons, which talks about making thresholds and just defining them. And as long as we're transparent about the rationale and the criteria we're using, which those tables show, reasonable people could argue about it, we could change them, but we're going to come to some answer.
Fantastic. We'll take one more quick question.
I have to say this is the talk I was waiting to hear for a very long time, so thanks for that. I was wondering, this situation is, I guess, relevant not only in nature conservation, but also in other fields of society, etc. So I'm interested in like whether there's like a social science, scientific roots to like choosing aid, decomposing it that way, etc. Something about like, the evidence behind that this is like the way to go about.
I've got one hidden slide that I was going to talk to this. I'll go really quick if I could.
Quickly.
It's not going forward. Okay. So this is what I call the gold standard curve. And what it shows here is that we're making investment in evidence acquisition and analysis for all our decision making. This is the probability of making the right decision. If we don't do any science or research or evidence acquisition, we might get lucky and make the right decision. Chances are low. If we do a bit more, we're going to get better. We can get to the top here if we do gold standard research and all the kind of high-end stuff that academics and our PhD committees want us to do. Then we kind of start to lose some steam, and then we get to planning paralysis, okay? The thing is, for the vast majority of decisions, we don't need the gold standard. You actually need the aluminum standard, which is here. It's quick, it's actionable, it's recyclable, it's fast. And for most of our decisions, we only need to get here. Part of my problem in the practitioner world is I have all these academics telling me, well, you're not doing the gold standard, so therefore what you do doesn't count. But the answer is not this versus this, it's this versus nothing. And so we have to get to those actionable points.
Fantastic. Excellent. Round of applause while Philip comes up. Okay, so next speaker is Philip Borsch-Suppan, and he's from the British Trust for Ornithology. And for those of you who don't know, this is an amazing organisation which for about 100 years has been a non-campaigning group that uses science to protect the future of birds by both inspiring people and informing decision, providing the science to inform decision-making. So over to you, Philip.
Excellent. Thank you, Julia. And she just gave the first page of my talk, so that makes it easier. It's a pleasure to be here. I'm neither British nor American, really. but both countries have been a big part in my life, and yet sometimes I feel a bit of an outsider. And I look at Britain, and one of the peculiarities I notice is that there's a real passion for collecting in British culture. And I think it's not just collecting objects, it's collecting events also. And I think really that is what has given us this really rich history of participatory science in the UK, because things like bird watching, things like collecting data on other taxa, really have a firm place in the public. And so the BTO is a small organization. We're about 150 staff. But we estimate that in the last year alone, we received volunteer efforts worth over 2 million hours, or the equivalent of having about 1,300 full-time field workers. And that's amazing to work with. I mean, that means we can do things like having these national species assessments and also do targeted conservation work. And so one of the flagship schemes we run is what's called the Breeding Bird Survey. And there have been about 9,000 people involved in the last 40 years of this. We're currently monitoring about 4,000 sites, which covers in excess of 1.5% of the land surface of the UK. So this is kind of an amazing survey. It allows us to track the population status of about 3/4 of the breeding bird species, land bird species, that is. And so those trends cover the whole range from species that are in near terminal decline, like the turtle dove on the bottom, colonizers that are rapidly expanding, like the Chetty's warbler on my side of the slide. And then a lot of species are kind of doing all right, going up and down a bit. And these trends, they actually become government statistics in the UK, and they feed into policy instruments that are being used to evaluate progress against things like targets under the CBD or in domestic law, as Sally was talking about this morning. And I think they have a use there because you can answer questions like, are we halting biodiversity loss with them? But I would argue that these indicators are not sufficient, really, and perhaps also in sometimes not even necessary to inform making the decisions that we need to tackle the biodiversity crisis. And so I don't want to lessen all of the talk we had today about gaps, but I would argue that kind of we need to focus perhaps a bit less on the state of nature and perhaps a bit more on informing decisions. And I'm going to talk about two examples from our work where we're trying to do just that. And the first one is about protected area efficacy. And this is work that is underpinned by the Breeding Bird Survey and other general purpose monitoring schemes, and where we're trying to assess whether or not designation alone in the UK has a positive impact on species. And we found that that was the case when we look at the occurrence of species in protected areas. More species occur in protected areas than outside of them. They're more abundant in those areas, but we actually don't see that the trends in the protected areas are any better or not. And so there's some doubt about whether protected areas actually have a long-term positive effect on changing or halting biodiversity declines, rather. And getting to this point is not exactly easy. So these are observational data. They were not designed to assess protected areas. And so protected areas are not designated at random. So we used a lot of statistical modeling to try to actually get this efficacy measurement out. And in particular, in part of the analysis, we used a method called statistical matching to create a database counterfactual by pairing the protected sites with unprotected sites that had similar characteristics in geography and land use. And that really kind of narrows down even a really rich data set like we have in the BBS, because the the BBS designed to give these country level insights, but protected areas are relatively rare in terms of land cover. And then also they target specific habitats. And so for some things like wetlands, we hardly have actually any counterfactual sites that are unprotected. And so while we did get like some ideas of what works, what doesn't work, and using some demographic data we had, we got a hint on perhaps why the abundances are higher. So for a subset of these 100-odd species we have in the left part of the slide, for about 20 of them, we can see that generally unprotected areas, they're more productive. But yeah, that doesn't seem to translate into population growth. The second example I want to talk about is about a single species, and that species is not doing particularly well. The black-legged kittiwake, this dapper gull, has halved approximately in the last 40 years. And the reason I'm talking about a single species is because the regulatory context that we have when we look at energy infrastructure development in this case is by and large based on doing single species assessment. So we have a framework where the regulator is interested in the integrity of the protected area network and their features, which tend to be single species. And that's not easy. So kittiwake populations are being pressured by multiple pressures. There's fishing, there's disease, there's climate change, and now there's offshore wind, which may impose further threats. And while we do have trends of the population, it's unclear what the relative impact of these different threats are. And so, for example, even kind of 25 years after building the first offshore wind farms, we know extremely little about the extent of collision mortality for those birds. They're long-lived, so it's really like it takes a while for any intervention, for any threat to be picked up in these population trends. And so in this infrastructure sector, almost all of the assessments that we do, we rely on modeled counterfactuals. So we don't have data, they're wide-ranging species, we cannot kind of pair sites that are affected or not affected. And so we rely on mathematical or simulation models and expert judgment. And really all I want you to take away from this slide is this is a mathematical model of a particular intervention of introducing artificial colonies. And we have data on this productivity parameter demographically. We don't have data on this dispersal parameter that's necessary to know if the birds come back to the population. But there's a plausible space, parameter space, where this intervention will never work. And so we don't know if it will work. But these things have been consented at large scale, and I think it wasn't the intent of the regulator to create an experiment here, but we have one now, and I think it's up to us to monitor and see if this works or not. And so I want to conclude by kind of pointing out some ideas of how we overcome this gap between the monitoring of the state of nature and actionable information for interventions. And I think we've heard a lot of this today already, so I'll keep it brief. I think we need to integrate beyond those state measurement schemes. So we need to have targeted interventions that are structured such, targeted monitoring, sorry, that are structured such that the data we collect is interoperable with the long-term monitoring that we're running. I think we need to think more mechanistically in how we set up counterfactuals when we plan interventions or planning decisions. And I think measurements alone will not solve the biodiversity crisis. So like we really need to foster collaboration and communication between policymakers and those who measure, because there's, I think that disconnect between state of nature measures and intervention monitoring mirrors a gap between these high-level international biodiversity ambitions that are on this abstract measure of biodiversity and national legislation that still tends to be single species focused. And I think in that communication, we need to be honest as the measurers what we can and cannot measure, because if policymakers create targets that are based on quantities that we cannot measure, then those policies will fail. And so we need to be honest what we can and cannot measure. And if we need quantities we cannot currently measure, then we need to think hard about how we can change that. And I think technology is on our side, and we've heard a lot about that today. And so I want to end here. Thank you.
Fantastic. Thanks, Philip. Questions for Philip? One quick one, probably. Go ahead.
I'm kind of curious, looking at the data that you shared with the protected versus unprotected areas, what the differences in stewardship of those sites are.
Yeah, that's a really good question. We did actually do a follow-up study. The information on the state of protected areas in the UK is a bit hard to interpret, but we generally found that those sites that were deemed to be in a favorable state actually had positive trend outcomes. A really big challenge is how that favorable state is defined. And England, which is the largest constituent country, has a state measure that turned out to be non-interpretable for this purpose. So we know that favorable or management that results in favorable status works in Wales, Scotland, Northern Ireland. We're less sure about England. But we still then, it's even harder then to turn specific actions to what generated this favorable state. But yeah, and I mean that reflects other work like Hannah Walcop's work, that management is needed, and like designation alone will not get us out of this. It's quality of protected areas.
Fantastic. Round of applause for Philip while I invite Kat up. Thank you, Philip. Thank you. So Kat Bruce is the founder of Nature Metrics, which is a tech company that's trying to make biodiversity measurements scalable and talking about giving people the best day of their life. She recently gave Prince William the best day of his life. I hope it was, again, the best day of his working life, when together they sprayed pond water at journalists while she was demonstrating how eDNA data extraction works. So, Kat.
Thank you. Yeah, I think I peaked that day. Thanks so much. It's such a pleasure to be here. I was a biodiversity scientist, never in a million years meant to be a tech company founder. I was working on DNA metabarcoding back in 2012, 2013, when we first started pulling that tool set of next generation sequencing out from the microbial world and applying it to larger organisms. And I was really struck by the extent to which we were all writing papers that concluded by saying this was going to transform the world of environmental monitoring and solve all of these long-standing challenges. And yet no one was really taking it outside of the academic research sphere and making it available and accessible to those who are at the front line of doing that. And so I took that jump back in 2014 and have been cutting my teeth on how to do this in the real world since then, through a time in which the general understanding and urgency of addressing biodiversity has been really transformed. 10 years ago, I went to investors and said we were going to revolutionize the scale of biodiversity monitoring. And they were like, what? Who's going to pay for that? No one's interested in nature and biodiversity. So it's a very, very different world that we live in today. We have quite a big team based in the UK. We have a lot of PhDs, which has its pros and cons. But that ranges from, we've got ecologists, data scientists, bioinformaticians, molecular scientists, all working together on how we can crack some of these sort of big challenging issues. That's this. Okay. So in a nutshell, we take DNA from water, from soil, and starting to work on DNA from the air as well. We sequence it and turn it into data right the way across the tree of life, from microbes to megafauna, from nematodes to newts. There are lots of alliterative ways of saying that. But generally, generating species data at scales that just has never really been possible before. And that's transformative because it allows us to move away from having to make a priori decisions about what we think is going to be interesting or useful or possible to measure. And we can actually just measure very broadly and see what is changing and see what is responding. And it allows us to take more of a big data approach to biodiversity, which is by its nature a big data challenge. It's vast and messy and complex and noisy, but there is signal within that noise that can tell us whether things are broadly getting better or worse. So we take those enormous data sets and turn them into a plethora of different types of metrics and indicators and measures and descriptions, some of which go up, some of which go down, all at the same time. We can do lots of ecological stats to look at changes in alpha and beta diversity across time and space. We spend a lot of time doing survey design to make sure that we're going to get the right sort of data to answer the questions that we're doing. We can look at what species are where. And that was all great when you're working on very well-defined projects, which are run by ecologists who are used to dealing with all of that sort of data and synthesizing it and putting it into reports and doing whatever they normally do with it. One of the contexts that we started working on in this sort of very project-focused way was companies doing environmental impact assessments, particularly in sort of mining, energy, infrastructure contexts. They have to implement the mitigation hierarchy. They have big teams of ecologists who are working on impact, on baselining, impact assessment, monitoring restoration, etc. They write these huge reports with masses of spreadsheets, all sorts of different types of data. They're measuring different things in different places, which you would want to do because every context, every scale, every project, every habitat is different. And then they send their reports to somebody centrally who's who's responsible for biodiversity, and also to a lot of this is about license to operate within the jurisdiction, so project funders and the countries. And then that data didn't really go very much further. The people sitting above the person who's responsible for biodiversity in these companies, until quite recently, had never really thought much about biodiversity. And then along came things like CSRD, TNFD, SBTN, all of these acronyms, and suddenly CFOs and chief sustainability officers are having to report to investors and to consumers and other people in their supply chain on their biodiversity impacts and dependencies. Which panics them a little bit, and they do not know what to do with all of this stuff. It's way too much, it's way too different, they don't want p-values, they don't want 1,000 lists of species, they don't want any of that. They want to know, are we on track? Is it good or not? Are we better or worse than our competitors? And are there any major business risks? And if you're not careful, we leap very fast to very simplistic measures that are just, what country am I in? What's my area? What industry am I in? And it's utterly unrelated to what's actually happening on the ground. And the problem is, on the ground, you've got stakeholders to whom that really local, very specific outcomes are really, really, really important. So you end up with a massive amount of distrust because the people at the top are ticking boxes and the people at the bottom are like, that doesn't reflect what we're seeing actually happening. So what we were really trying to do is go, look, the people at the top need simple measures, sure. The world of AI allows us to take really complex data and use that to drive really simple measurements. So how can we start doing that within this context? So we need to start making the measurements, the metrics that we're using aggregable so that they can be summarized up. There might be a lot of complexity, a lot of different measures here, but can we combine them up into something that summarizes where we're at? And we need to make them much more scalable. So a couple of examples of how we're doing this. This is I haven't actually put what it's called. This is our restoration tracker product, which is using underlying sort of beta diversity analyses. You need to have a reference site. So in this, you need to have something locally where you go, this is an example of what we're trying to create, a good quality habitat. And then essentially what we're doing is, this example is with soil. So we've taken soil samples in the restoration sites. We've analysed them for bacteria, for fungi, for invertebrates, so 3 different big separate data sets. And we're saying both compositionally and in terms of their functions, how similar are the communities we have in our restoration sites to the ones that we have in our target habitat. And that allows us to put a quantitative score on a scale from 0 to 1 on how well we're doing at restoration. For the people at the top, that score is all they need. But that score is an average of a score for every sample in your site, and the score for every sample is a combination of the responses of the bacteria, the fungi, and the invertebrates separately in their compositional and functional states. So you can disaggregate this into all of that data to see what's responding, what's not, what's doing better, what's not, which taxonomic or functional groups are underrepresented in your restoration areas. And all of this information is what you need to inform the management so you can optimize the outcomes as quickly as possible. So you've got the information for the people working on the ground, and it's the same information that's then being summarized up into those simple metrics at the top. Incidentally, this is soil. We have also done this, actually I think it's on the example, this example, which is from work we did with the Forestry Commission, where we were sort of putting this into action in sites which some of which had been just started their reforestation journey, and others that had been being reforested for a couple of decades. In this example, yes, we used the soil, but we also did bioacoustics and generated bird species lists, and we were able to use the bird species in exactly the same way here, and then see which functional and trait-based groups were missing from the restoration sites. The challenge with that is that you need the reference site. So not everywhere has a good reference site. Who determines what's a good reference site? You need to keep monitoring the reference site, which is expensive. It's not massively scalable. So in the UK, we now have a lot of soil-based data through work we've done with the Scottish Government, with Forestry England, with some agricultural projects and various things. We've got enough data to start building a model-based approach. So here we're able to take the sites where we have soil data, overlay that with geospatial data on habitats and land use, and then combine it with something like the BII, the Biodiversity and Taxonomy Index, to build a model that lets us take any single soil sample and say where it sits on a scale from good to bad based on its biological community, and also lets us do things like species distribution modeling to predict where important species might be, even in places where we can't go and monitor them. So this is just an example of applying that model-based approach to two sites. Again, NEP is sort of the gold standard rewilding example in the UK. It's been sort of 20 years since it transitioned from farmland, and comparing that to a new site that's just starting to be rewilded. So bringing that back to the sort of what companies are looking to do, that's getting us some of the way to being able to aggregate things up, provide some simple metrics that underneath them embody all of that complexity and are driven by it, but it still needs boots on the ground. And a lot of companies are not even at the stage of knowing where their locations are, let alone having people there who can send in samples. So what we've been working on now is really starting to scale this vertically. So looking, if you're coming top down and you're a company coming to biodiversity early on, how can you go right the way from the top, the TNFD, LEAP assessments, etc., understand your portfolio, pull out information on that, use geospatial data to understand your habitat metrics, use that combined with existing data to do species distribution modeling to look at your most high-risk sites, use all of that to then start doing plan your surveys for ground baselining in the most important sites, set your KPIs and monitor against them. And at the sort of center of all of this really is the idea that you should be able to have a set of site-based KPIs that are relevant to your site but are aggregable, and underneath them contain all of the information that the people on the ground can trust. We have an opportunity, to really make a difference here. We need radical collaboration between stakeholders who have no trust, no common language, very little history of working together. Data can become that common language that can help build the trust if we can use the same data to package in different ways to meet the needs of the different people in that stakeholder group. So, thank you.
Fantastic. Thanks, Kat. Quick questions from anybody. Go ahead.
Thank you, Kat. That was great. I'm Lauren Alexander Augustine. I run the Gulf Research Program here at the National Academy. And I have a crude question. And I'm wondering, what is your budget? What does this cost?
It depends. It depends what you're trying to do and at what scale, to be honest. Yeah, it's sort of low hundreds of pounds per sample, which is to unlock that whole community from the samples. So sort of low thousands of pounds to really start to getting to an idea of what's going on within an area. And then it depends, are you setting up essentially a full experimental design to answer a particular question, in which case costs do become quite high, quite fast, or are you combining, are you doing a purely model-based approach, which will give you information at a different scale, or you combine those things to sort of spread costs over long time.
Let's talk.
Fantastic. Go ahead.
Such a great talk. Thank you so much. I guess my question is, what is the baseline, especially from a temporal perspective, and sort of going into KPIs, because that will fit into KPIs. So what baseline are you working with? Because the baseline is always shifting, right? Mm-hmm. What are we dealing with here?
So in the specific example of companies who are doing environmental impact assessment, for example, generally your baseline is project before the project starts. So what's the impact that operation is having? As you start moving to a more general measures of ecosystem quality or ecosystem condition, then it's imperfect, and all we can work with at the moment is the best available information that we have. I think we always know that our top score is going to be probably not pristine. In fact, we definitely know that it's definitely not going to be pristine, but it still allows us to say broadly, are we getting better or worse, and where are we? Where are things better or worse than others? A lot of the value is relative, but yeah, we'd absolutely expect there should be some places where you could get more than 100 based on the model.
Fantastic. Thanks, Kat. And next up, we've got Janine Cavendish-Bars, and she's Professor of Organismic and Evolutionary Biology at Harvard, and also the Director of the Harvard Herbaria. And she's done absolutely foundational work on plant functional traits. So over to you, Janine.
Thank you, and thank you so much for the invitation to be here. I'm going to go back to, how do I advance this? There we go. What Andy Purvis said this morning about biodiversity being complex, hard to define, and hard to measure. And this is connected with the challenge that the National Academies got from the National Science Foundation in the US to think about continental scale biology and how we do that when biological processes are operating at many scales, and that biologists have divided themselves into isolated disciplines where we each measure one thing at a time, and we don't have good ways to integrate across the different scales. And so there's a need for continental scale biology where we work across scales. So Scott Getz and I were both on this committee. Cliff Duke is here, was an organizer, and Jack Liu was the chair of this committee. And I worked mostly on the tools. And one of the advances in the tools for capturing biodiversity is remote sensing information, as we've just been talking about in the breakout group. And this work has been advancing, and there's been a huge investment in this with the National Ecological Observatory Network in the United States, with NASA investing in their surface biology and geology mission. I'm sitting next to Amy Rosenthal from Planet, learning about Tanager, the European CHIME mission. And Scott and I worked together to create this figure for the National Academy's Continental Scale Biology report that's just coming out. And thinking about the ways that we can use hyperspectral and LIDAR, remote sensing, other kinds of remote sensing to measure all kinds of phenomena above ground that also allow us to predict processes below ground through different kinds of AI models. But it requires a lot of investment on the ground with biologists really understanding what they're measuring and then connecting that with remotely sensed information. And another kind of information that we heard about this morning from Daru is the wealth of plant diversity information that is housed in the world's herbaria with over 400 million specimens. And I'm now directing the Harvard University Herbaria, which has one of the largest collections in the world. And we have just launched the International Herbarium Spectral Digitization Working Group, and we met a week or two ago in Cambridge to bring enthusiasts, directors, curators from the world's herbaria together to develop protocols and standards for using spectroscopic information, so reflected light from herbarium specimens, to predict a whole suite of plant functional traits. So similar to the kinds of things that Daru was trying to do, we can predict a whole suite of functional and chemical traits from fresh or dried, pressed leaf tissue that are housed in specimens using machine learning models, as you see here. And this is a wealth of information that's really critical for trying to scale up plant functional trait information to global scale. We can couple it with other kinds of trait information housed in trait databases or collected from airborne data where models are used to predict functional traits at large spatial extents, or global maps from satellite data. And we can build these and connect these kinds of upscaled maps with other kinds of functional trait maps that can be built from remote sensing. And so for the past 5 years, I've been leading the SEND Biology Integration Institute, funded by the National Science Foundation, advancing spectral biology in changing environments to understand diversity. So we use spectral information, spectroscopic reflectance across scales, from leaves to tree canopies that we might get from drones, or airborne data that we might get from NEON or other platforms, and satellite data, and try to use that as a common data type to integrate biological information across scales. And one thing that has come out of this is an understanding that spectral information is coupled to the plant tree of life. And when we look at spectral signatures of plants that you see in the bottom left, so these are spectral reflectance signatures of different plant species from leaves, from fresh leaves. And these are the wavelengths on the x-axis, the reflectance values in each of those wavelengths. And we look for different major clades of vascular plants. We see different parts of the spectrum that show high phylogenetic signal, meaning that close relatives have very similar spectral signatures. And across the tree of life, from all the data that our group put together, we show that spectral signatures are really more similar among close relatives. We see that at the leaf level, but then when we take the NEON data, we also see that when we take pixels, where we can identify an individual pixel to a specific tree canopy, and doing this across 21 NEON sites, it doesn't render very well, but we basically see the same thing. Unfortunately, those graphs are not rendering, but they show where you see the red dots, and these are the wavelengths on the x-axis. We also see phylogenetic signal in pixels, spectra in pixels, remotely sensed from airborne data. And this is at the leaf level. This is an airborne data from NEON. This is in the forest and biodiversity experiment. This one is at Cedar Creek in Minnesota, where I set up this experiment, and we find that these spectral signatures, even at the canopy level, are also linked to phylogenetic information. And we can use the spectral information across the NEON sites to model multiple measures of diversity, functional diversity, phylogenetic diversity, species diversity, and using different Hill numbers, we can get at the dominant species, or we can get at evenness, or we can get at richness. And all of that can be well predicted from spectral information. This is with sub-meter, 1 meter spatial resolution. Gets a bit trickier when we try to think about the satellite data that will be at 30-meter resolution. And so this is getting ready for the surface biology and geology mission. But here we're using DESIS data from the German satellite that's at 30-meter pixel resolution. And this is EMIT, US satellite data that is 60 meters. And this is what that data look like. And we can also do some dimension reduction using wavelet transformation with that. So we have now taken the forest inventory analysis data. This is a major investment, the U.S. Forest Service, we're very worried that this investment might be under threat. But there are hundreds of thousands of plots across the United States that have been monitored regularly for every tree stem that's over a centimeter in diameter. And we can create ordination. So it's a dimension reduction process where we can get axes of ordination. The ordination are based on distance matrices that could be using species information or phylogenetic information or functional trait information. All of those give us, in this case, 3 different axes that we can then couple with spectral information because it's geolocated that we get from satellite. And so here are results of these models using the DESIS data at 30 meters, but it's 400 to 1,000 nanometers, or EMIT, that's 400 to 2,400 meters, but it's 60 meter pixels. And we get pretty decent predictions of where each community, each forest community is in that 3-dimensional space. And if we look at the variable importance in projection, this just shows which wavelengths are important in making those predictions using machine learning models. We see that the 2 sensors show really good overlap in the wavelengths that are important in those predictions. So even though they have different sets of wavelengths that are used in those models, which wavelengths are important are consistent between the different sensors. So we can use that to actually build maps of taxonomic community composition. So where's your mixed hemlock beech forest, the phylogenetic composition, the functional community composition, or we can drill down and we can look at taxonomic composition and actually where's your black spruce forest, your upland forest, your sugar maple beech birch forest, and then create these composite figures that map out different forest community types. Or we can take a phylogenetic approach, find where are the pinus species more likely to occur, or the beeches or the maples, and make composite maps that map out those different phylogenetic groups of trees. Or we can do it in terms of functional traits. So we can build these maps that give us different kinds of diversity information. And now we are taking on the task of using herbarium specimen traits to build those ordinations, which give us the advantage that we can get functional trait variation through time, not just at one point in time. We can get functional trait variation latitudinally across climatic gradients so that we get much more nuanced functional trait variation, and we can use those to build the ordinations that we then predict from satellite data. Why is any of this useful, or why does it matter? So at the COP16 meeting, which was truly a joyful, exciting meeting with over 20,000 participants, we were all talking about how do we measure biodiversity and how do we use it to meet the global biodiversity framework targets. And for these particular targets, this kind of predicted biodiversity information can really get us to understanding of ecological integrity, or how well we're doing with conservation through time, because these are all updatable maps that can be predicted through time as long as we have the in situ monitoring information to validate that information. We can track invasive species. We can look at restoration efforts and see if they're actually working. And so these kinds of tools are really critical for the biodiversity monitoring to feed action and for verification of the work that people are trying to do. So I'll just stop there and take questions.
Thank you very much, Janine. Questions? I'm going to go for Kamal at the back there.
Really terrific stuff. Are you able to detect loss of function? You refer to functional diversity in some of your work. And the second question is, to what extent the spectral signatures are different of herbarium specimens and the live specimens?
Mm-hmm. So are we able to detect loss of function? So we're able to link functional trait information or reflected photon information to ecosystem functions. It all depends on being able to measure those things on the ground. When you say loss of function, it depends what function you're talking about. One of the most measured functions that ecologists take on is productivity. So we can measure declines in productivity. We can measure how biodiversity is connected to productivity and how changes in biodiversity are connected to changes or declines in productivity. If you want to talk about air pollution removal, for example, if that's the kind of function you're talking about, we model that, we can model that, but it depends on the data going into those models actually being accurate. And so if we see declines in forest cover and leaf area, that would be a loss of that kind of ecosystem function or ecosystem service. I'm not actually sure which kind of function you mean, because that is a word that can mean many things.
That sounds like a further discussion. That's fantastic. Great. Okay, I think we're going to go straight on to the next speaker. So thank you very much, Janine. Great. And our last speaker, although you don't get a break after this, so that doesn't really help, is Anil Madhavapedi, who's Professor of Planetary Computing at the University of Cambridge. And one of the things he's famous for is taking amazing notes from meetings he attends and then making them publicly available. So Anna, we need to hit him up for those notes. And I also read on his bio that he really wants to see a pika, the small mountain mammal in the wild. So if anyone can hook him up, that sounds like one. There you go.
I am most excited about seeing a pika in real life. Thank you. So it's great to be here from the Cambridge Conservation Initiative. I think these are a slightly old version of my slides, so if I follow a sync, I apologize. But what I wanted to do was to go back to where we started today's session on, which is when Andy Purvis told us that there's no one way to measure biodiversity. And really, it should be the case that we shouldn't try to create a single metric for something that is fundamentally really quite a complicated and very kind of multidimensional field. So it is a little bit strange whenever you think about how we deal with machine learning, that we're trying to, in many, many cases, construct a single kind of perfect model that will form the basis by which we make lots and lots of complicated decisions. So the exciting kind of frontier that I wanted to talk about was a journey we've been going through to try to understand how we can get to multimodal AI, which is a connection of models that can all learn something quite specific and quite deeply about a field, and then interact with each other and to try to form kind of a more meshed network of these kind of models, which can then in turn inform hopefully high accuracy implementations of biodiversity decisions. The journey for all of this began with a project I began with my colleagues in zoology and plant sciences and geography about 3 years ago. And the idea behind the life metric was to pick a specific actionable metric for land use changes. So if you're about to convert a piece of land on the planet to either from natural use to human use or vice versa through restoration, then what is a comparable cost for this conversion around the world? And the specific one that we chose was to measure the probability of species extinctions and so on. And then the reason that computer scientists got involved was because of the huge amount of data involved. So there's a whole bunch of rasters from multiple species, so 30,000 species. And in total, the amount of data we manipulate is around 7 or 8 petabytes of data. And so this is something that used to be almost impossible. It's now pretty straightforward, but it still requires a certain amount of specialist kind of data processing knowledge to kind of work through that much data. And the idea was that if we can make this kind of a very transparent pipeline, then we can make it much more defensible when policymaking figures out the trade-offs between these kind of things. And so this just got published in Philtrans B earlier this year. Now, the basic mechanism for life in the beginning did not use any AI at all. So the idea is to create an algorithm which is a fairly explainable algorithm. And then from the basis of this, we should be able to then figure out how to make it more and more high resolution. So in a nutshell, the idea behind LIFE is that we take expert maps per species. So if you take the pika, for example, we take the IUCN red list information and all the subspecies of pikas, and these are usually hand assembled by experts. And then from there, we can calculate the areas of habitat that represent, by combining these with elevation maps and other kind of raster data, where these things are likely to live. Not an STM, but a binary kind of area saying that they're there or they're not there. And from there, we can then calculate per species the chances of extinction if one of the areas of habitat disappears. And then we can aggregate all of those to a massive addition and come up with a single map that kind of tells us if you were to go from a particular set of species living on a single tile and you convert them away, then what is the aggregate extinction risks for all of these species there as well? So pretty straightforward. But the problem is that when you build one of these maps, you build it at a certain resolution. So in this case, LIFE is built at 1.8 kilometers squared, around an arc minute. And when you zoom in and look at the map, you can see all kinds of interesting artifacts. So you can see, for example, suspicious circles where the exact kind of species range wasn't known. And in this case, the colors represent red for mammals, blue for birds, yellow for reptiles, and green for amphibians. But the artifacts tell you exactly what the scaling limitations of this thing are. And so the natural temptation with AI is to say, well, we can take this and we can just predict from a model what a high resolution version of this is going to be, based on some land use predictor or something. But the problem is we're not really giving it any more information. We're kind of guessing at this point. To get to the ground level, to get to the next level of resolution, we need loads more information in order to understand the nuances of what is actually happening on the ground. And with this, we can probably get to some like a 70 or 80% level of accuracy when we drop to the ground. But we really want to get much, much, much closer to 100% before this is a useful thing for reliable policy making at the local level. So the basic approach is to think, well, we've really got two different modalities of information. If we can kind of use them both and construct reusable infrastructure for these things, then we should be able to combine these things in order to get better information. And so the two things I want to quickly talk to you about is the idea of using observations, which are basically things that you see from remote sensing from space. And then a completely different modality is what we know. So the combination of the world literature that represents just centuries of human endeavor of running experiments and also of indigenous knowledge, where things are recorded about causal facts that we've put together. I'm going to first start off with observations to give you a sense of what's going on there. Then I'll describe the same for literature, and then I'm going to put together a little challenge for us about how what we need to do in order to move forward with this properly. So the idea behind observations is using something called foundation models. The basic problem with satellite data is that it is ginormous. So we've got petabytes and petabytes of data from just one instrument, and we have many, many more birds going into the sky with lots and lots of diverse instruments. So the idea behind the machine learning approach here is to build something known as a foundation model. And what a foundation model is that it's a general model that's been trained in this data that has no specific downstream purpose. Instead, it's a reusable artifact that can then be tuned for many, many downstream tasks without having to be retrained. The initial training is the computationally enormously expensive bit, and the more downstream tasks you can find that are useful, the better off we go. Without going into too many details, we took all of Sentinel-1 and Sentinel-2, so synthetic aperture radar and multispectral data, and we had just one insight that made this model particularly outperform, which is that we pass over the same bit of Earth many, many times. And most often, it's cloudy in interesting parts of the world like tropical rainforests. And most of the time, most people just remove the clouds. But instead, if we just stack all of the cloudy images into one megapixel that we call a D pixel here, we get lots and lots of fragments of information from those repeated passes. And so given that information, we can then distill down lots and lots of the data into a spectral temporal model. So that is one that doesn't know where it is, but for each pixel on Earth, we can figure out what, across the course of 1 year, what the spectral signatures of this thing are. Now, these numbers don't really have that much meaning. They're just a very compressed representation of what is down there. And then from that, you can start to visualize the representations. If you do dimensionality reduction, you can see that across the whole planet, we can see colors in false color, where similar things like fields, for example, or mountainous areas or rivers and so on, they all have different colors in different parts of the world, but the representations are starting to match. So we can start to use this to get a sense of how to group spatially similar things with similar signatures for things like crop classification, land segmentation, canopy height estimation, and all kinds of stuff. The crucial thing is that once you train one of these foundation models, we then have a spatial modality that can be fine-tuned with relatively small amounts of data. And so each of these cases are very, very diverse, but all we have to do is to put the expert knowledge from each of these areas into a much smaller training process in order to get results. And so, for example, crop yield analysis is surprisingly straightforward to train with one of these things. And this whole foundation model was trained on around 8 GPUs, so it's not a Google scale kind of problem. So it also gets really fun when you start combining these kind of foundation models. So in this case, we then took some of the spectral models, and then we combined it with hyperspectral sensing to start using the models in the domains that they're really good at. So in this case, for example, we used a Landsat-based optical segmenter to figure out the canopies of tropical trees, and then a hyperspectral pass that went through multiple times to do species identification across multiple tropical trees. So this kind of combination in this one domain is proving really, really quite effective in order to put these together. So this is one foundation model trained based on satellite data. What if we switch entirely and say, okay, we've hit the limits on what we can do by looking at things from space. We can do similar things by looking at your ground data, but ultimately there's huge data gaps, as we've learned today, in what we can see through any kind of observational process. So if we turn to the literature, we start learning causality. We start learning the whys of things. And so in order to make this work, Bill Sutherland approached us with a problem in conservation evidence where they've had a team working for a couple of decades on manually doing systematic reviews to show that there's good testable practices for conservations and then recommending those actions. And this, from a machine learning perspective, was a heaven sent because it gave us a labeled data set where humans had looked at this thing very carefully, and then it let us use that as an anchor to benchmark ourselves against a much wider effort. And so then what we did was, well, why do we have to just stop with the papers we manually download? So we went to the University Library in Cambridge and we said, can we just download all of the papers? And they said, well, I mean, how many is that? I said, no, just all of them. Can we just get them all, right? And then we found one librarian who had an encyclopedic knowledge of every contract that Cambridge had ever signed with every publisher. And he decided he wanted to help us with this. And so we downloaded 100 million full-text papers after much drama. And then we can create a pipeline where we can just learn across the entire body of conservation papers. And we have a pipeline where you can then cluster these things. You can run them through evidence pipelines. You can figure out what the relevant papers are for some inclusion criteria. You can then really examine them carefully through larger language models. So use just the right language model for the thing you're doing, categorize a smaller set, and then pull out semi-structured data from the papers. So you can say, is this testable action happening in a completely in a specific region of the world? Are the results kind of measurable on certain axes, and what are the methods involved? And then it also spits out a nicely categorized and annotated PDF, so a human can visualize exactly what is going on when they verify this thing. So the AI is not coming up with results, it's simply classifying kind of what's going on, and then the humans in the loop kind of figure out what's going on. So this stuff gets really fun when you start looking at the visualizations in the same way that we looked at the visualizations to the spatial stuff. So this represents around 10 million papers or so. And what's going on there is that the video is not playing. Let's see. I think I might. How do you go back here? If we can go back and just see if we can play the video. Oh, it's playing now. So what we've done here is we've tried to map papers into a metric space. And so what we're doing here is that every green paper is one that has been in a journal relevant to conservation evidence. And the other ones, the gray papers, are ones that have not been looked at or are ruled as out of scope. And so you can see here that we can already start clustering papers just by looking at titles and abstracts into kind of a metric space. And you can start to see the relevant kind of connections between them all. And then what you can do is you can say, well, can we extract out a much richer knowledge graph about how all of these things connect together by looking at the full texts and kind of topic areas and so on? And at this point, there's lots of hidden layers in the model. This is an internal representation of what the model kind of looks like. Each of those clusters, they still represent papers that have been looked at by conservation evidence or not. But you can see that behind the scenes, it's starting to look almost like a living organism. There's lots and lots of hidden connections that we're still exploring to try to kind of form the causal connections between these things. So what we now need to do is to figure out, well, once you put these things together, we now have a pipeline by which you can connect all the dots together. So you can say that we can take multiple modalities, and then we have humans in the loop at all times. And then we can continuously reconstruct these things in order to ultimately create products from lots of these data sources that are quite good. But I wanted to highlight, just to close, some problems that might bring all of this crashing to a halt. The first one is that if you ever feed back in data that has been generated through AI sources, it leads to something called a recursive model collapse, because you're training something on something that doesn't have any genuinely new information. And similarly, we're seeing that in the literature from 2023 onwards, there's lots of AI-generated papers. All of these things are combining together to mean that we might be in a situation now where the use of AI and the lack of tracking is resulting in our inability to get more training data in order to progress some of this research. And so moving forward, we need to do a couple of things. The first thing is that it's vital that we figure out what the provenance and reproducibility story for some of these data sets are. We've heard today loads of exciting stories about how different parts of biodiversity can be tracked, classified, and so on. We have to keep the outputs of AI models separate from anything else. And the second one is that we need some competition in order to keep some data secret, so we can essentially have exams for AIs. So every year, we're currently doing the equivalent of setting an exam, say for MADS, and then repeating and reusing the questions every year. So obviously, some models will accelerate ahead because they already know what the questions are. What happened in protein folding was that experimental biologists would discover proteins and keep them secret for a year. And then every year since 1994, they've had competitions, which then allowed the computational predictors to kind of move forward. If we can figure out collectively how to do that in biodiversity, it gives us that kind of meta framework in which we can plug all of our data and our learning and our models together, and then maintain a healthy competition where we can learn from each other and then cooperate and try to figure out exactly how we can push this forward. So thank you very much for that.